are you? Hello. Okay. You're live. Everyone, no one can hear you, but you can hear me, so there we go. All right, so Chapter 5 is all about energy. When you think about human metabolism, a lot of what you think about in human metabolism is the breakdown of glucose into energy. That's sugar, right? Now, we do know there are other ways. Think back to your digestive lecture. There's other ways that you can get energy as far as, fats, carbohydrates, proteins. That's the same as far as microbial species go. But one of the big differences that we look at when we're looking at how we produce energy is that microbes can do a couple of things that we cannot do. They are actually more efficient than what we are when it comes to looking at exactly how they make their energy. And so we're going to kind of be focused on the microbial aspect. So even though some of it is going to be very similar to what you have heard, as far as eukaryotic species and your own metabolism goes, you have to understand the title is the title of the chapter is microbial metabolism. So it's going to be different than what you learned in A&P. All right, so when you look at metabolism and what that is, it's talking about chemical reactions, summing everything up. How do we build things up? How do we break things down? Remember talking about building things chemically. If you take very small particles, like glucose in the body, and you put a bunch of glucose together to build up this large molecule of glycogen, that's a reaction that is called anabolic. You take something small and build it much larger. On the other hand, if you take that glycogen and then break it down into the individual glucose molecules, that is called catabolism. And microbial species are fantastic at taking things and recycling them. We do not waste anything as a microbe. They will take things up, they will build things, they will break things back down, and anything that they can recycle, they will do so as not to waste something. When we're looking at making energy, the process of making energy is critical. They are having to survive cellular division. Even though they don't do mitosis, they still have to divide, they still have to reproduce, they still have to have an energy form. And so utilizing these chemical processes in a microbe is very similar to the importance that it's going to be as far as your body going. Remember when we talked about in A&P the fact that microbial metabolism and taking a look at, human metabolism, and taking a look at why we need energy, looking at the chemistry of the body. It's the same when you look at microbial metabolism. They have to have chemical energy as well, and there has to be some source to provide the necessary either carbon source or the necessary supply to make their ATP. When we look at a couple of properties in microbial metabolism, we do have to understand that many of the reactions happen much faster than human reactions happen. Understanding metabolic pathways allows us to really understand then how microbes are working. And when we can understand how microbes are working, we can understand how to control them from a human aspect. So understanding the properties of making their ATP and providing energy allows us to be able really to control them. And that's what, from a human standpoint, that's what we're wanting to do. We want the good microbes to stay, and the bad microbes we need to figure out how to get rid of. Microbes do utilize enzymes. Now, we talked about enzymes in A&P, so let's just kind of think back to what an enzyme is. Enzymes are biological catalysts, which means what? What's that mean? Faster, right? Happens faster and with less energy. Those are the two big things. If I am going from point A to point B, let's say after this class, I am taking off to Florida, because I am. Okay, just saying. I'm not trying to rush you guys. And let's say that I decide I have two options. I am going from point A in Kentucky to point B in Florida. And that does not change, right? Destination is the same. But let's say I have two options how to get there. I can either jump on a personalized jet, which I don't have, but whatever. Jump on a jet, take off, and land in Florida, what, hour and 45 minutes later, right? Or I can ride a tricycle all the way to Florida. Do you know how long it would take me to ride a tricycle? Can you imagine? Like, what the? Now, am I still getting from point A to point B? Yes. Which one is faster? Obviously the jet. Which one uses less energy? The jet. Can you imagine having to cycle your way, and on a tricycle, not even a bicycle, right? Like a kid's tricycle. Can you imagine how long it would take you to get there? Vacation would be over before I could even get there. I'd quit long before. I'd be like, done. I'd get maybe to Clarksville. Be like, actually, I changed my mind. Our jet is an enzyme. It gets us there faster, and it utilizes less energy. And enzymes are very specific. We don't have destination point A and B when we're doing chemical reactions, but we do have reactants and products, right? I have a reactant, I'm going to kick out a particular product. Reactant, product, reactant, product, reactant, product. That's what we do. In order to kick out that specific product, we have to have a specific enzyme. So they're a lock and key method. The enzyme doesn't fit just any old substrate. It has to be the substrate that fits in what's called the binding site. So there's a specific, and it's basically based on shape, right? So if we have the correct shape of the binding site, we can we can bind the substrate and kick out a product. Okay. We also know that we can control, to a certain extent, how fast or how slow chemical reactions can go. I can manipulate a chemical reaction, change the temperature, for example. One of the reasons why your body turns up the temperature just a little bit when you get sick is because things move faster when the temperature is turned up. Your body's trying to heal itself. I can manipulate it based off of particle size. Remember, the smaller the particle, the faster the reactions go. Think about kids. They're little and they're fast. Turn around for 2 seconds and they are gone. And they're not gone like 2 steps away. They're like down the block, already inside somebody's house eating supper with them. And you only looked away for 2 seconds. Because they're little. And they're fast. So smaller particles tend to create faster reactions. So we can manipulate, to a certain extent, how quickly chemical reactions are actually moving. When you look at a chart of an enzyme, the black line that you are seeing there, that is a reaction without an enzyme. And the kind of pinkish magenta line is that exact same reaction, but with an enzyme. So it happens faster and it utilizes less energy, which is something that we, as microbial species or humans, want to happen. When you look at enzymes, you make those. Microbial species make these. And one of the things we have, let me turn this so you guys can see the board. Whoa. Oh, by the way, I do have a black eye because my son and I were pretending to be on a roller coaster and he headbutted me. Just letting you know. So if I have an enzyme, there's a couple of things about this enzyme that I want to point out. It is made of a protein portion. There's a non-protein portion. But this right here, that site right there, that's called the binding site. That's where a substrate has to bind. So let's say that this is the enzyme. And these are the substrates that are available. Which substrate, 1, 2, or 3, would fit into that enzyme to kick out a product? Number 2, why do you say that? Because it matches, right? That's the shape that would fit in that enzyme. So this substrate is going to bind to that active binding site, and it's going to kick out a product. And that happens over and over and over and over. So when you look at it up here, this substrate that we're looking at on this example that you're seeing, it's binding there to that active binding portion so that it can kick out some kind of a substrate, or I'm sorry, some kind of a product. When we get into looking at how we make energy, we're going to talk about some of these important coenzymes that we have, that we make this way. But we'll do that in just a second. One of the things that I will say about enzymes, they do turn over very, very quickly, which means they're going to bind a substrate, boom, kick out a product. Bind a substrate, boom, kick out a product. This can turn over up to 10,000 molecules per second. That is a lot. Think about doing anything 10,000 times per second. When you look at that, did I tell you guys that Kendall planned an impromptu sleepover? Because they had practice this morning. So this is the little girl's mom wanting to know, hey, what time is it over today? So I'm going to tell her so that she doesn't come back to my house. That's awful. I love her. So we kick out these products. Now, remember when we were talking about enzymes in A&P, one of the things that we said about an enzyme is they do absolutely have to be in the tertiary state to be active, which means wound up in that three-dimensional shape. Remember, there's the linear, that's primary, not active. Tertiary states, where they're wound up in that three-dimensional shape, those are active, and therefore we can use those. Enzymes, therefore, can be manipulated. I can manipulate an enzyme to be active or to be inactive. And sometimes that might be reversible, meaning I can make the enzyme inactive and then turn it back to activated. Sometimes it's not reversible. It's irreversible. For example, let's say that David gets up and I say, what do you want for breakfast? Number one, I don't ever cook him breakfast, so this is a fairy tale. So I say, what do you want for breakfast? And he says, you know, I think I want scrambled eggs. So I say, okay. So go over to the stove, crack the egg, right? Start scrambling. It's cooking. I'm singing. And he says, you know what? Changed my mind. Kind of want over easy eggs. That's the runny eggs, right? Now, scrambled eggs are already almost done. Can I change that? Egg is a protein, right? That's what the yo nasty yo double was going to be a little baby chicken. Crazy. Anyway, scramble that up though. How did I manipulate that protein? What did I do to it? How are you cooking it? You add heat. I put it on the stove. I don't put it in the refrigerator and start scrambling, right? No, I added heat. Heat denatures proteins, which means it takes those three-dimensional bonds and breaks them, making them linear. Now, sometimes I can take that linear species, remove it from the heat, and it will go back into activated form. Not always, just like that egg. If I take that egg off of the heat, I cannot then make it into over easy eggs. You were eating those scrambled eggs because that's what you said you wanted. It doesn't go back. I can change the pH. pH has a huge difference. Too basic or too acidic, I can manipulate enzyme activity. I can also manipulate how fast the enzyme is working to a certain extent. It is called saturation. If I want an enzyme to be working, then I have to have substrate available. If this number 2 is not available to this enzyme, am I going to kick out a product? No. The more of this substrate that is present, the more product that I can kick out, right? To a certain extent. Let me show you this chart. Take a look at enzyme activity. As we increase substrate concentration, which means I'm adding more and more and more substrate. So I'm going to take number 2, I'm just going to start dumping it, a lot of it. Enzyme activity rockets up. Do you see that? Then it plateaus off. Why? Exactly. Because eventually, you're going to hit a point when if I freeze time, that enzyme is always bound to a substrate. It's always kicking out a product. That enzyme can only work so fast. So dumping more substrate really doesn't do anything once it's reached what's called saturation. So I can manipulate enzyme activity by increasing the levels of substrate to a certain extent. Then you're just kind of wasting substrate. Another thing we can do, controlling this enzyme could be by inhibitors. And there's 2 types that we're going to look at, competitive and non-competitive inhibitors. So the first one I want to talk about is a competitive inhibitor. When you think of a competitive anything, what do you think of? Like a race, a competition, right? Somebody needs to be the winner. Anybody really competitive by nature? Nobody? Really? Okay, I am bad. And what's even worse is so is David. I mean, like, the kids will be like, Mom, do you want to play basketball? And I'll, like, demolish them. I'm like, boom! Beat you again. Not really. No, don't do that. But it takes all I have not to. But now, when me and David play, like, you know, golf together, or, we're out for blood. It's a serious thing. And it might not even be like that. I mean, Kendall has said before, like, Mom, it's not that serious. I'm like, but it is. My kids don't have that competitive drive. I don't know how do you not when both David and I do. But that's actually the first thing. My sister, when she saw my eye, she said, oh, geez. Marie and David, like, playing basketball or something. I was like, no. That was last week. No. Because, I mean, we're just competitive by nature. Well, a competitive inhibitor does manipulate or alter enzyme activity because of a competition. This is the substrate, yes? What if I had in presence, though, something that looked like that? Is that the same? No. Is it close? It's very close. Just this one little piece is different from the original. And so what can happen is this competitive inhibitor can jump into that active binding site. But when that happens, am I going to kick out a product? No, because that's not the substrate that should be there. So I took that active enzyme and actually made it inactive, right? Most of the time I can reverse this, which means if I can add enough substrate to flush out that competitive inhibitor, the enzyme will go back to working. If I can remove the competitive inhibitor from that site, the enzyme will go back working. However, sometimes these are irreversible, which means if this competitive inhibitor binds, that's it. That enzyme is done. I cannot reverse the process. So if you look up here, there is the normal binding substrate that is going to kick out a product versus the competitive inhibitor, which looks quite a bit like the substrate, but am I going to kick out a product if that is present? I'm not. So that enzyme would be inactive. In a real-world example of this, now don't worry about focusing on, this is organic chemistry, but what you're seeing up here is a sulfa drug. You've heard of sulfa drugs, right? Antibiotics. Anybody allergic to sulfa drugs? There's usually somebody. Okay, I am. Take for example, though, that you go in and your physician is going to prescribe for you an antibiotic, including sulfa. If you look at this here that says PABA, don't worry about the minor details, but PABA is a part of a chemical reaction that ultimately gives us folic acid. Microbial species have to make folic acid if they're going to survive, period. There's no exception there. Look at the two organic structures. They look very similar. That core structure there is actually identical. It's the side chain that's different. So what they do is when you take a sulfa drug, that competitive inhibitor jumps into the chemical reaction in place of PABA. So therefore, do you make folic acid? No. And if you don't make folic acid, bacteria die. That's how that drug works. So the reason why when you take an antibiotic on day 1 and you're not, you don't feel any different, day 2 you're like, oh please, day 3, day 4, now you're like, all right, I'm finally starting to feel a little better. Because when you take that drug, it doesn't just automatically wipe them out. It takes a little bit of time for them to exert their effect, and then the microbes start to die out. So that's an example of a competitive inhibitor. A non-competitive inhibitor is also known as what's called an allosteric inhibitor. And the reason why it's called an allosteric inhibitor is because many times on an enzyme, there will be a separate binding site. It's not the site, though, for the substrate. It's a secondary site. Like that. It's called the allosteric site. An allosteric inhibitor will come in and it will bind to the allosteric site. But when that happens, automatically we get a shape change of the substrate site. Now watch this. Let me ask you a question. Looking at it right now, do I, in fact, have a substrate that could bind to that site and kick out a product? Yes, number 2. So that's an active enzyme because I have substrate that could bind. Allosteric inhibitor is going to bind to the allosteric site, and watch this. It changed the substrate site, changed the shape. Look at my 3 choices. Do I have a substrate that's going to bind to this new shape? Nope. Therefore, it was active, and I've now made it inactive. Can't use this. That's one thing that can happen. Now watch this. Let's say that this is the enzyme that I'm starting with. Do I, in fact, have any substrate present that's going to work with that enzyme? No. So that's an inactive enzyme. I can't use it. Allosteric inhibitor is going to come in and bind to that allosteric site. It's going to do a shape change. And now it's going to change it to that. Well, do I have a substrate that can bind to that? Yes. So then, I actually took an active and turned it inactive. Or I can take an inactive and turn it active, like that one. So even though it's called an inhibitor, it's hard for some people to wrap their head around the fact that we can actually turn an enzyme active. So let's look up here. Here's the original. There's our substrate. There's our active site. That can bind and can kick out a product. So is that active or inactive enzyme? Active, because I can kick out a product. Now I'm going to bind that allosteric inhibitor. Altered the binding site. Can that substrate now fit? No. So in this example, it was active and it turned it inactive. So those are all ways that we can manipulate enzyme activity. There's other ways, though, in the body that we can control things, like feedback inhibition. We talked about feedback inhibition in A&P, negative feedback. Remember where you build up your product and then you stop production. It's kind of like an assembly line. Or in my household, it's like grocery shopping. Does anybody like to grocery shop? Raise your hand. Anybody? Anybody? No one is raising their hand. I don't even know what that is. Shut your face. What are you saying? Kroger's pickup stop?
Yeah, you can go online, grocery shop.
You stop it.
And then you drive up and then come and pack your car up.
What?
Life changer.
I need a moment. Why do I don't think our Kroger does that.
It's called a clip stop.
Who goes and gets the groceries?
The waiters go and shop for you. They just like their child.
You gotta stop.
And then if they can't find something, then they'll switch it out for something similar.
I feel like at this moment my life's just like in my head it's like angelic sounds coming. I hate grocery shopping because of my kids and David. I know that's really mean, but they're terrible. I don't want to grocery, and I can't send David to grocery shop because he only gets like half of the items. So then I'll start to prepare a meal, and I'm like, where's the rest of it? He's like, well, I don't know where to find it. So you just stop looking, like, what? The kids are terrible. And they'll just like throw, like what? Stop. Running up and down the aisle. Stop. So for me, when I go grocery shopping, it is like an ordeal. I'm the lady with like 2 carts that fills it to the brim. And then I go home and I stock everything up. It's beautiful, right? Freezers stocked, pantry stocked, shelves, cabinets, refrigerator. Then what do? Well, you start using stuff, right? You start making things, taking ingredients, and your supply starts going down, right? Refrigerator starts to look a little bare, cabinets. You can't go all the way empty, because then the kids are like, are we going to eat food again? I guess. So you don't want it to go bone dry, but you do let your supply kind of dwindle, and you're like, all right, I'll go back to the grocery, and you replenish your supply. That's how feedback works, right? You'll build up this stock, and then you'll utilize what you've made so that you don't waste anything. And then you'll kick back in production. I got to check that. I will be looking at, what's it called?
It's Kroger's like
Kroger Click and Stop.
something like that. And then
I'm going to need you to be a little more specific.
I'm gonna have to look it up. after
Okay.
class.
Yeah, we'll be looking it up
So,
together.
but after you do it, like there's like a really quick survey at the end of it, and they'll give you like 400 fuel points to get your gas there as well.
I just gotta move on. Chemical reactions are happening in the body all the time. There's different types of reactions. And one of the ones that we're going to focus on is what they call redox, reduction and oxidation reactions. And that is where we're moving electrons, right? Something's going to gain an electron, something then is going to lose an electron. Gain, lose, gain, lose. And in the body, we focus on the major biological electron that is necessary for biological systems as being hydrogen. Hydrogen can be moved, for example, down the electron transport chain so that we can not make energy but utilize it. So we're going to start looking at, in fact, how do we make energy and how do we utilize it? ATP. Doesn't sound like much, doesn't look like much, but that's energy. It's an adenosine with 3 phosphates. If you only have 2 phosphates, it doesn't work as energy. It's got to have 3. An adenosine triphosphate, I'm going to sit, is made. You have to generate that. Now let's think in AMP. We talked about generation of ATP, right? Things like cellular respiration. Which organelle of a eukaryotic cell makes ATP? The mitochondria. Remember, that's called the powerhouse. Well, bacteria do not make ATP in their mitochondria. Why? Because they don't have it. They don't have organelles. So this is all happening not in the mitochondria. We're going to utilize just in the cytoplasm and on the plasma membrane for ATP production. When we look at how we can make ATP, humans do cellular respiration. There's aerobic and anaerobic. Microbes do as well. Cellular respiration is the process of taking glucose, breaking that down, and we either are going to do aerobically or anaerobically to make energy. If your final electron acceptor is oxygen, that is aerobic cellular respiration. If it's anything else, I don't care what it is, sulfur, nitrogen, carbon, gasoline, I don't care what I put. If the final electron acceptor is not oxygen, that can't be aerobic. That is anaerobic. The energy that you produce aerobically and anaerobically as a microbe is very different. Aerobically is where you're going to generate the most ATP. Very successful. Anaerobically, they can generate ATP. They're much slower in how they do it. They don't, it does not provide the same amount of energy. So we're going to look first at aerobic respiration. And one of the things that you kind of have to revisit and think about in your mind is the Krebs cycle. Now let's think together. You start with glucose, which is sugar, right? We're going to break that down into pyruvic acid, which then gets broken down into acetyl-CoA. And acetyl-CoA enters into the Krebs cycle. The Krebs cycle is a step-by-step different reactions chemically that's happening. And we're only going to focus on two things. I'm not going to make you re-know the Krebs cycle or go back and revisit all of that, but we're going to focus on two important parts of the Krebs cycle. So if we're looking where we're at kind of big picture-wise, we're going to start with glucose. We're going to break that down into pyruvic acid, which then gets broken down into acetyl-CoA, and that enters into the Krebs cycle. On the Krebs cycle, let me show it to you. It looks like this in its entirety. There are 8 different reactions that are happening in the cycle. The only one that I really want to focus on, though, is the succinyl-CoA to succinic acid. It's number 5 here. Why did I pull out succinyl-CoA to succinic acid? What do you see there? That's the step that we generate ATP. You've got to be able to make it. Critical. I also want to point out, though, some of the byproducts. Now, the byproducts on here, they're in this lavender purple looking color. NADH, FADH2. What do each of those end with? Which is hydrogen. Remember when I told you that the oxidation reduction chemical reactions that we were going to focus on, in fact, utilizes hydrogen as the electron to move. Well, that hydrogen doesn't just magically appear. We get that hydrogen as a byproduct from the Krebs cycle. So, these hydrogens that we're fixing to talk about in a second, they do in fact come from a byproduct of the Krebs cycle. Making ATP is really only part of the story. You must be able to utilize ATP. If you cannot release ATP to be utilized, what's the point of making it? For example, got up this morning, I'm the queen of crock pot, FYI. If I can use a crock pot, I will use it. So, got up this morning and put in a roast. Cut up some potatoes, cut up some carrots, put in some seasoning, mixed it all together, turned it on. Which is great, because then it's done. So let's say I go home this evening, crock pot's there, turns itself automatically right off. So let me ask you this, did I make dinner? Yeah, it's cooked, put it in this morning, finished. But now let's say that I just let it sit there. Never take it out of the crock pot, never put it on a plate, never actually eat it. Are we still hungry? Yeah. You can make dinner, but until you utilize it, eat it, who cares if you made it? My kids don't care if I cook, as long as they're fed. They'd probably rather I don't cook and feed them something ordered. Just because you make ATP, that's not the entire story. You have to be able to utilize it, which means we have to be able to release it. And that involves the electron transport chain. The goal of this, keep this in mind, the goal is to be able to release energy. And the process of doing this has a name. It's called chemiosmosis. So let's look at a big picture where we're at right now. Glycolysis is the breakdown of glucose. So we're going to take glucose, we're going to break that down into pyruvic acid, and over here on the respiration side is where we are at currently. So we're going to take that, enter it into the Krebs cycle. That's going to give us the byproduct that we need for that hydrogen. That hydrogen is going to enter into the electron transport chain. Goal is to be able to release energy. And in order to do that, the process is called chemiosmosis. So let's take a look. Let me pause 2 seconds. Any questions on where we're at right now? We're good? We're all together? You are where I am? People are like, okay, all right. So when we enter the electron transport chain, there are pumps found on this chain automatically, like the sodium-potassium pump, right? That's a pump found on the membrane. Okay, so there's pumps that are found on this membrane that actively pump hydrogen, which is nice because guess what electron we're using? Hydrogen. So we're going to take hydrogen and we're going to physically pump that across the membrane. When I do that, I start to generate two types of gradients. The first type of gradient is a concentration gradient, right? The second type of gradient is an electrical gradient. Concentration gradient, because now I have more on this side than the other side. An electrical gradient, because each time I pump across a hydrogen, that carries with it a positive charge. So this side starts to become more positive than that side. There's a channel right here that is controlled by an enzyme. The enzyme is called ATP synthase. That channel is closed until the electrical and the concentration gradient reach a high enough point. And we don't care really how high that number is, but the concentration and the electrical gradient will slowly start to creep up. And when they reach a high enough point, this enzyme recognizes that and will open that channel. When the channel is open, do you guys remember talking about equilibrium? That's a balance, right? Things move from high concentration to low concentration. Things move from positive to negative. So based off of that information, when this channel opens, the hydrogen over here is going to flood to the other side. Goal being to reach equilibrium again. But that flood of hydrogen, that rush through that channel, allows us to release ATP. Let's look at it up here. There it is. There's our, now this is on the, it says up there, mitochondrial membrane. This was a good picture from eukaryotic species. One thing I want to note with you, it's not on the mitochondrial membrane for prokaryotes. Bacteria don't have that. It's on the plasma membrane. Okay, so don't let this picture confuse you. I just wanted to show you the channels pumping that hydrogen across so that then we get the concentration and the electrical gradient. That enzyme recognizes an increase in the gradient. It's going to open that channel. Hydrogen floods through. That's what allows us to release ATP. That whole process has a name, and it's called chemiosmosis. Okay? Now, that is aerobic respiration. In prokaryotes, you generate 38 molecules of ATP. Do you remember how many you generate as a eukaryotic species? 36. It's less. There's actually an extra step that eukaryotes go through. They make 38, but there's an extra step where they lose 2 ATP. So they end up total 36. Prokaryotes, that doesn't happen. They make 38 and they keep 38. They have found a way to bypass that extra step. So really, when you're looking at who's more effective, who is more effective, prokaryotes or eukaryotes? Prokaryotes. They're very good at what they do energy-wise. That is not the only way that they can generate energy. They can do anaerobic respiration. If the final electron acceptor is not oxygen, I don't care what it is, but if it's not oxygen, then we can generate anaerobically. Anaerobic microbes are a little difficult to grow in lab. Think about it. What do you have to deplete if they're going to grow? You've got to take away their oxygen, which happens to be atmospherically everywhere, right? But we can do that. We can actually grow them. They're a much slower growing microorganism. You have to be a little more careful with how they grow. And they're much slower because they don't generate as much energy as what aerobic respirators do. So when you're looking at where all of this is happening, the big difference is the fact that prokaryotes don't have a mitochondria. So the parts of where it would be eukaryotically in the mitochondria, prokaryotically that is done either in the cytoplasm or on the plasma membrane itself. When you look at the energy produced, aerobically versus anaerobically, prokaryotically versus eukaryotically, prokaryotes generate more ATP than eukaryotes, and aerobic respiration is going to give us the most energy. 38 molecules for prokaryotes. Now, questions on that? Okay, so we're not quite done. We are almost done. Let me pause for 1 second. So the reason why we're not quite done is because microbes can actually utilize one other property for the generation of energy that we can't. And not all microbial species can do this. It does depend on what type of species do we have. Do they have the proper enzymes, substrates available? Are they cellularly able to do this? And for most microbes, the answer is no, but for some, the answer is yes. Another way that we can actually generate energy is through a process called fermentation. Fermenters can take that glucose, they can break it down into pyruvic acid. That's the same as what I showed you for cellular respiration. However, instead of then taking it and turning it to acetyl-CoA into the Krebs cycle, if they are fermenters, they will take that the pyruvic acid and turn it into either alcohol or lactic acid. Those are the two types of fermenters that we have. Has nothing to do with the Krebs cycle. Has nothing to do with oxygen. Doesn't really care about the final electron acceptor. But it also does not give us very much energy. Microbes could sustain this way for a little bit. So let's say this happens. Let's say that I have an aerobic species. That means I have a bacterial species that has to have oxygen if it's going to survive. Yes? Yes. Now let's say I take away its oxygen. I deplete it with something like cyanide. That's what happens, that's how people die from cyanide. It depletes all of the oxygen, so they die. If that microbial species needs oxygen in order to live, and I take away the oxygen, that species is dead. It cannot all of a sudden become an anaerobic species. That doesn't work that way. But if it can ferment, then it could switch over to fermentation and survive by fermenting for a little bit. Now, if oxygen never comes back, eventually that species would die. It's kind of like a reserve tank. Have they ever been scuba diving? I like snorkeling, but not so much scuba diving. I'm a little bit claustrophobic, and I don't like not being able to see behind me. Like, if a shark's going to eat, you know, it's like, oh, look at the beautiful fish. Shark kills me from behind. Like, no, I don't really like that. But when you go scuba diving, they give you your air tank, and then they give you a small reserve tank. The reserve that you can switch on is not so you can stay down there for hours longer swimming around. It's so that you can finish what you're doing and get back to the top because you're out of time. So the reserve tank for these microbes is fermentation. Little bit of energy can live a little bit longer. We talked about fermentation when we talked about Louis Pasteur, right? When we were looking at pasteurization and how those certain microbes can actually ferment alcohol. That's what we were looking at. Depending on which microorganism you're talking about, though, some of them do, in fact, give lactic acid. Well, why would we want to know this? Here's why. Remember, as a microbiologist, your goal is going to be to figure out what unknown microbial species am I looking at. First thing we do is what? What's the most important thing? We're going to gram stain it. You're going to tell me, hey, is it gram negative? Is it gram positive? What shape is it? Because remember, that eliminates everything else. But then, I have to start doing tests to figure out what I actually have. And one of those confirmation tests that we can do is called the fermentation test. Sometimes I want to know, is it a fermenter? Because if that answer is yes, that's going to send me down one path. If that answer is no, that's going to exclude all those fermenters and send me down another path. So many times I just want to know, is it a fermenter? Does it ferment alcohol? Does it ferment lactic acid? And by answering that question, I'm a little bit closer sometimes to what species I do in fact, have. So you will have to be able to understand and interpret the fermentation test. So, in the fermentation, it's not difficult. Actually, it's very, very simple. Fermentation test tells me two things. Number one, do I have a fermenter? And you'll answer yes or no. And number two, do I have a gas producer? Sometimes microbes release a byproduct of gas. It's usually carbon dioxide. I need to know that. Test is simple. I take a test tube that is full of liquid. Let me turn this screen to make sure they can see the full board here. All right. So I'm going to take, okay, maybe I should just stop. I'm going to take this test tube, and inside of it I have a small test tube inverted, upside down. That's called the Durham tube. There's going to be liquid inside of here, filled to the top. Inside of the liquid, mixed up, there's one protein, there's one carbohydrate, and there's a pH indicator. pH indicators pick up changes in pH. Did it go basic? Did it go acidic? And they change colors, right? Think about, remember doing litmus paper, like in chemistry, it would change colors based off of, blue or pink. Do you have an acid? Do you have a base? Well, this liquid is usually some kind of phenyl red substance, but it is a red, kind of bright red liquid. And that pH indicator is going to pick up on whether or not we have changed the pH levels, and if so, there would be a color change. And it's very easy to see the difference. If there's a color change, when you come back and look at this test tube, it's no longer red, it's yellow. That word right there drives me insane. My mother says yellow. She says it all the time. Yellow. What is yellow? Yellow. How do you spell it? Y-E-L-L-A? Yellow? Stop it. Yellow. Anyway, random. I'd look at it. If it's yellow, that tells me there's been a change in pH. Second thing I'm going to look for, it's straight observation. Is there an air bubble present in this Durham tube? And it would be a large air bubble. Sometimes you'll get kind of these little bubbles in it. That doesn't count. These air bubbles are large bubbles that collect. And so you'd see a big space of air at the top of the Durham tube. And that's it. Like, that's what you look for. But you have to understand why. Microbial species will only choose one source of carbon. They will not choose both. I have given them two sources, one protein and one carbohydrate. They'll have to pick one or the other. So I'm going to take a bacteria and I'm going to inoculate that, which means what? That's a fancy word for put, right? I'm going to put that bacteria into this solution, inoculate it, and I'm going to let it grow. I'm going to incubate it. It's going to choose one source of carbon, one source of energy. If it chooses the carbohydrate, not all of them can choose the carbohydrate. If it chooses the carbohydrate as its energy source, then what happens, as a byproduct, it releases an acid. Well, the pH indicator is in there to pick up on changes in pH. And is an acid a change in pH? Yes. So we get a color change to yellow, confirming the fermenter. So when you come in, you would look and say, yep, that's yellow. That's a fermenter. But what you would know about that is it utilized the carbohydrate as the energy source. A lot of times on the exams, what I find is people second guess themselves. It drives me insane. Because they'll be looking at a question and like, ooh, I know this. Ooh, I know this. The answer is a fermenter. It's a fermenter. So they look at A. They look at B. They look at C. They look at D. And none of them say, is a fermenter. So then they start questioning themselves. Well, I guess not. Then they just pick something randomly. Well, I guess I'll pick A. Wait a minute. Because if they would have read D, it says, utilize the carbohydrate as the energy source. What is that? That is a fermenter, right? So being able to correlate that information together is important, that you understand, not memorize this. Understand what's happening. That's key. That's how you learn information. If you can really walk your way through it and understand what's happening, then you know it. Not if you can memorize a sheet of paper and vomit it back to me. So when you understand what's happening, the reason why I have that color change is because I use the carbohydrate as the energy source, ignoring the protein, created an acid as a byproduct, pH indicator picked that up. And the other thing we look for is just whether or not that air bubble is there. If the answer is yes, then it produced a gas. If that Durham tube is filled to the top still with liquid, then the answer is no. So there's really 4 things that you can get from this fermentation test. You can have a fermenter and a gas producer. You can have a fermenter and a non-gas producer. Those do not, just because you're a fermenter doesn't also mean that you're a gas producer. Then you can have a non-fermenter, non-gas producer, or a non-fermenter gas producer. And again, it's to help us identify these organisms, because I know which species are fermenters and gas producers, which means everything else is eliminated. So let's take a look at a test, an example. Take a look here. These have all been incubated for at least 24 hours. We'll say 48 hours. Is A a fermenter? No. Why do you say that? Because it's red. It started out red. It ended red. Which means, what did it use as its source of energy? The protein. Can't be the carbohydrate, or it'd be yellow. What about D? Is it a fermenter? That's an obvious difference, right? Big time color change. We also then need to look if it's a gas producer. Take a look at A, B, and D. All of those are gas producers. It may be hard to see where you're at right now, but there is an air bubble that's pretty large in the top. Now C has some small air bubbles in it, but if you look at it, there's still yellow liquid all the way to the top of that Durham tube. Which means it is a fermenter because it's yellow, but it is not a gas producer because there's not that big air bubble in the top. So I did this test for us, right? Taking a look at these 2 test tubes. Is this a fermenter? No. I know that because it's still red. If I look inside of it, there's no gas bubble. So this is a fermenter. non-gas producer. What about this one? Obviously yellow, right? So it is a fermenter. Again, there's no gas bubble in the top of that though, which means it's a fermenter, but not a gas producer. So A or B, which one utilized carbohydrate? That one, right? So understanding the fermentation test is really just being able to understand kind of what's going on within the test tube itself and being able to understand why that's happening. Questions on fermentation?
Is that the, not the anaerobic, but, yeah, is it part of the anaerobic?
It is not. This is separate. Not all species can ferment. This is completely separate from cellular respiration. This is taking glucose into pyruvic acid, and then we're going to ferment that into either lactic acid or alcohol. And not all species can do that. So it is separate. Then it doesn't have anything to do with the final electron acceptor like what we were talking about with cellular respiration. It doesn't have anything to do with the electron transport chain or the Krebs cycle. Nothing. This is completely separate. Just another of course, obviously we can't ferment as humans, right? But a separate way that they can actually make energy. So that is actually the end of material for test 1. So you now have chapters 1, 3, 4, and the material from chapter 5 that we're going to cover on exam 1. See? Not too bad. It's exciting. Your faces scare me. Your faces scare me.